The prompt for this section asks to explain how a mutation is formed through an insertion or deletion by giving the protein sequence of the normal and mutant chain. The normal chain protein sequence is methionine, tryptophan, phenylalanine, threonine, lysine, and glycine, and then the chain continues. The mutant strand sequence is methionine, tryptophan, isoleucine, histidine, and then the chain stops after that. Using the coding dictionary developed by Marshall Nirenberg and Philip Letter, we can find all of the possible mRNA sequences that code for each amino acid. From the dictionary, we know that the only possible mRNA sequence for methionine is AUG, and the only possible sequence for tryptophan is UGG. Phenylalanine has two possible codons, UUC and UUU. Threonine has four possible codons. The first two letters are AC, and the last letter of the codon can be ACU or G. Possible codons for lysine are AAA or AAG. The codon for glycine begins with GG and can have any of the four mRNA nucleotide bases for the last letter. In the mutant strand, isoleucine and the stop codon can have three possible codons, while histidine only has two possible. The normal and mutant strands start out the same in the beginning since both have methionine and tryptophan. There is a change from phenylalanine in the normal strand to isoleucine in the mutant. This indicates that the mutation, which is an insertion or deletion, occurs in this area. If we look at the possible mRNA codons, we can see that isoleucine starts with an A, but there is no A in phenylalanine. This suggests an insertion taking place here. After adding in the A to the normal strand, we can start figuring out what the exact codons are for each protein. The first two bases of phenylalanine are UU, so isoleucine would have the codon AUU. Since histidine begins with cytosine as the first base, the last base of phenylalanine is C, which makes the codon UUC. We can go through all of the possible codons to match normal and mutant sequences. Part of lysine in the normal sequence changes to the stop in the mutant sequence. Therefore, we do not know the last base of lysine and the exact codon of glycine because the mutant sequence stops. Once we determine the exact codon for the normal and mutant sequence, we find that the reading frame has changed. This means that the insertion led to a frame shift mutation.